Hello, I'm Dr. Ogilvie. I'm speaking with you from my office where I will be answering emails, answering posts on the course website, and also interacting with the course website, posting quizzes, grading quizzes, etc., etc. So I just wanted to spend a little time so you could greet you and welcome you to the course in histology, where you are going to learn the microscopic architecture and function of the human body. I wanted to take a few minutes to share how histology was taught in the past. And in order to do that, I'm going to take the camera and I'm going to take the mirrored off and move it over to my other screen and show you what a histology lab looked like in 1963. I first came to MUSC in 1965, but actually came down here 2 years before and was involved in research. And by 1965, I became a part of the university and was completing my doctorate, as I completed in 1968. Well, this is what the lab looked like back then, and each student had a microscope which they had to purchase or rent, and some could afford a binocular, that is 2 eyepieces, and others could only afford a monocular or 1 eyepiece. And they had a box of slides that they would pick out and study, and there was a manual that guided them in studying that. And then when we gave practical tests, we would use their microscopes, and they would be out of the room, and we'd put a slide at each station, and we might have as many as 50 stations, and then they would all line up, and we'd give them a minute or two to look in there and identify something and write it down in a blank, maybe numbered from 1 to 50. And that was called a practical exam. And then the faculty would get together and use a key, and several of us sitting around and grade those practical exams. So now let's get this camera back up here and change back to the mirrored. And then this is a microscope that I had when I first started out. And then gradually over the years, it wasn't too long that I was able to afford and use a microscope like this, both for teaching as well as research. But now, everything's virtual. The slides that you study in WebMic were captured by taking photographs through the microscope at different levels and nesting the images and keeping track of where they are. And then I spent a whole year on sabbatical in the University of Zurich working with Dr. Peter Grosskreutz, who is my co-author of WebMic. He actually conceived of WebMic, and then I joined him, and we brought it to the next level. And then over the years, it has progressed until it's now an internet available resource that's free. There's a server at MUSC that serves it, and one at the University of South Carolina, and it contains lots of specimens, and you can read all about that in the WebMic section of the course website. And the other resource you'll be utilizing would be virtual slides or whole slides, whereas WebMic has snapshots. This is an example of aorta glass slide, and then a stain with hematoxin at the top, and then below that is a silver stain, an elastic stain to stain the elastic membranes that are in the aorta that you're going to learn about. And then below that is a liver that's very pink and almost square, and that's stained with hematoxin eosin, and below that is liver specimen stained with silver stain to stain the fine reticular fibers that form a network to hold the delicate liver cells. Now those slides came out of a box, which was the last box that was a loan collection for the students. And this at the top, it says Tissue and Organ Loan Collection. And you can see it's a box of about 100 slides. And those boxes were issued and checked out 1 to every 2 students. And they used those slides with a lab manual to guide their study. And then, of course, we would use some slides to test on that. So you will also, in addition to WebMic, you will be studying specimens, and where a special scanner was used to actually scan and reproduce the entire specimen and put in a pyramidal file so that everything is there in the algorithms on the server from which you will be accessing the specimens in BioLucida. Only what you ask for will be delivered to you. And so it's possible to take a 2,500 square foot image that resides on the server and only be transmitting what you're asking for. And so that's the way virtual slides or whole slides work. And it has enabled us to really learn histology, share histology, and even today, pathology is becoming very digital, and throughout the world, many pathology departments and in hospitals are going virtual to whole slides, and it's becoming very much like radiologists have been doing for years, where they can read a CAT scan or MRI or an X-ray from their home over the internet. So in the course, I think you will find adequate guidance on the course website. I will respond to any email, usually the same day. If I don't respond right away, just send me another reminder. But the best way to communicate about questions you have in the course are the forums that I've set up, where, like in the syllabus, the first page you come to, there's a forum where you ask questions about the syllabus. And I've tried to place those forums throughout the course website. It's much more efficient if you ask questions there. And if you need to remind me that you posted something, you're not getting an answer, just send me an email and say, I post something at a such and such place, and I will go there as soon as I can and answer it. It's much better if you post on the course website, because if you post and I answer, then the rest of the class will be able to get benefit from that. And it also saves repeating the same question and answer over and over. So I wish you all the best in the course, and hope that as the course goes on, we'll get more acquainted virtually through our interaction and learn histology. And I hope it goes well.